In this video, you will learn about the reproduction of a chicken, structures of DNA, the basis of reproduction of animal species, along with the shellless culture.
Reproduction of a chicken. First, to be able to fertilize an egg, you will need a rooster and a hen to mate. Hens lay eggs regularly, fertilized or not. It is only when the rooster's sperm enters the egg inside the hen, it is only then that the egg will become fertilized.
Is shellless culture possible? We believe shellless culture is possible due to experiments we have taken and multiple people before us. Shellless culture is the fertilization and growth of a chicken without a shell. We created an artificial shell with the minerals and vitamins of the shell.
Shellless Culture Journal. This is our daily journal of our observations, findings, and thoughts and ideas. Throughout the incubation and growth of the chicken embryos, we viewed the chicks during lunch at school to see them. Each day we had a class discussion about the experiment and our observations.
Day 1 of shellless incubation. Today we removed the eggs from pre-incubation. They were only day 2 of their 21-day incubation time. They weighed about 2 to 4 grams less than before pre-incubation. We cracked them into the culture vessel and prepared them for incubation. 3 of the eggs were cracked successfully, but 1 got broken. There are 2 out of 3 that have shown no sign of development. The other embryos developed right on schedule with blood vessels and heart forming and beating. That embryo has a coffee lid on it and 4 holes in the cup. So far we are amazed at what we are seeing.
Today, Day 2 of shellless culture incubation. Today we came into the classroom to the incubator dropping below temperature, which caused an alarm to go off. We are concerned it may play a factor in the embryo's death. However, we checked the developed one, it still has a heartbeat. We have ruled out the other 2 embryos as dead. The temperature has regulated at lunch to the proper temperature.
Day 3 of shellless incubation. The incubator's alarm was going off again, telling us the temperature was too low. We have come to a conclusion that the overnight temperature is the problem. Due to the school heaters being turned off at night, the room is getting too cold for the incubator, including the heat lamp to balance it, as soon as people start showing up and the room gets warmer. The incubator warms up with us. We have decided to add another heat lamp to our incubator to help keep temperature stable.
Day 4 of shellless incubation. The incubator temperature is still dropping at night, but not as badly as before. It regulated way earlier, and we took a look at it. We saw signs of development of the body. We have no true way of telling whether the chick is alive or not. We had an idea to create a smaller incubator to allow the temperature to regulate easier. We are very fearful of the upcoming weeks being 3 days, and the school heaters being off for all the time. On a good note, we are now able to see a body and a small eye.
Day 5 of shellless incubation. We have decided to put our plan into action and put any viable egg into a smaller incubator because the temperature is still dropping at night. We are putting 2 eggs in the smaller incubator. One of them is the well-developed, but not to the extent it should be. We think the growth has been stunted by the temperature drops. We aren't sure anymore about the survival of these embryos.
Day 6 of shellless incubation. We have ruled the embryos as dead. We are not sure why, but we think they could have been dead on Friday. There are so many causes of death that are all equally possible that we are not sure which one is right or wrong. Therefore, we are still trying to decide what the cause of death is.
Look at the lab report to find out anything that you aren't sure about.
Scientific method. The scientific method is the process of experimentation in order to answer a question. For example, scientists use the scientific method to discover how different types of species interact with each other. Follow these steps on making and understanding, learning the basics of scientific method.
DNA. DNA stands for deoxyribonucleic acid. It has a structure much like a ladder. We call this a double helix. The rungs of the so-called ladder are made of 4 nitrogen bases: adenine, thymine, guanine, and cytosine. There is a sugar phosphate backbone, which is the up rails of the ladder. These join with something called a hydrogen bond. Adenine always joins with thymine, and guanine always joins with cytosine. None of these intermix at all. A gene mutation is the alteration or substitution of one of the four bases in the rung. For example, adenine would switch out and become cytosine. This would cause a mutation. A gene is usually where the mutation occurs. A gene is a small segment of DNA that only holds a specific part that is needed. You'll find genes hidden in chromosomes inside the nucleus of a cell. A mutagen is any substance that can cause a mutation. There are certain mutagens, such as X-rays and UV rays, that cause mutations. Mutations are anything from Down syndrome to a freckle on your face. Mutations can be positive, negative, or neutral. A positive mutation will help a species survive in its natural habitat. A negative mutation will affect it and will not allow it to survive as well. A neutral mutation, such as being albino, will affect it in neither a positive nor negative way.
Genes. Genes are small segments of DNA that help to make proteins. This is done by chromosomes found in the cell of an organism. Everyone has the same gene structure, but if someone were to look closely at the basics of the gene, they could see the difference between them. Genetic traits. Genetic traits are things we inherit. This is a list of some of the traits that you could inherit from your relatives. Height, eye color, blood type, nose/ear shape. We can inherit other traits from the environment we live in. This might be to make our body more suited to our living conditions. Some of these traits are language, music we enjoy. Genetic traits can be easy to see, like hair color, eye color, and height. Others can be quite different to see, like asthma, musical talent, and ability to inherit a sickness. Genetic traits can be things you want and like, for example, to be tall or to be good at sports. Unfortunately, genetic traits can also be things you do not want to get, like cancer. Cells. Cells are the basic building blocks of all organisms. Cells can replicate independently and consist of many biological products inside of them. Cells contain cytoplasm, a liquid substance flowing inside of the cells, delivering nutrients to all different parts. In this section, we will take a look at the cell's reproduction using mitosis, cytokinesis, and meiosis. We will also look at animal cells, plant cells, and all their differences.
Meiosis. Meiosis is a kind of cell division that only happens in the production of a sex cell. One diploid cell will become 4 haploid cells. In this, there are 2 stages of meiosis, meiosis 1 and meiosis 2. In meiosis 1, the cell becomes 2 diploid cells. In meiosis 2, the diploid cells split into haploid cells. Mitosis. Mitosis is a cell division that has 5 stages: prophase 1, prometaphase, metaphase, anaphase, and telophase. In mitosis, the cell will prepare its nucleus into 2. Then it prepares for the big division, which is also known as cytokinesis. In prophase, the DNA condenses and organizes into a chromosome shape, which is more like an X. Prometaphase, the microtubules will reach out and start to latch on to the chromosomes. In metaphase, chromosomes will align in a straight line so that when the cell splits, it is evenly aligned. In anaphase, when the chromosomes split in half. Telophase, the membranes form around the 2 sets of chromosomes, forming 2 nuclei. Now the cell is ready for cytokinesis. Cytokinesis is the splitting of the cell.
Reproduction. There are 2 types of reproduction, asexual and sexual. Asexual reproduction. Only one parent is required in asexual reproduction. This means asexual reproductions reproduce offspring that are genetically identical to their parents. Some different kinds of asexual reproduction include binary fission, budding, fragmentation, and vegetative reproduction. Sexual reproduction. In sexual reproduction, a female gamete cell is fertilized by a male gamete cell. Gamete cells are created in the process of mitosis. Each has only one set of chromosomes. During the process of fertilization, these two cells will combine to form a zygote. The zygote receives half of its chromosomes from its female parent and half of it from the male parent. This means not two individuals will have the same DNA. The expectation is twins. The zygote will then go through mitosis and cell division to become an embryo. There are two types of fertilization, internal and external. In external fertilization, an egg cell and a sperm cell combine outside of their parents' bodies. It is very common in aqua animals. Internal fertilization is when sperm cells are deposited inside the female's body and meet the egg cell there. Most land animals use this method of reproduction.
Plant reproduction. Plants reproduce sexually and asexually. When plants use sexual reproduction, they produce offspring by the fusion of gametes, pollen, and falls onto the plant. This results in the offspring being genetically different from the parents. Some plants can produce a new individual without the fusion of gametes. They do this through asexual reproduction. The new organisms are generically identical to the parent plant, except when mutations occur. In seed plants, the offspring can be packaged in a protective seed, which is an agent of dispersal.